If a good sampling of sequences is the number of sequences is and the motif structure is, it should in principle be possible to obtain frequencies highly representative of the same motif in other sequences also. A. Available sufficiently large, not too complex. B. Unavailable sufficiently large, not too complex. C. Unavailable sufficiently small, not too complex. D. Available sufficiently large, too complex. The correct answer is available sufficiently large, not too complex. The quality and quantity of information provided by the PSSM also varies for in the motif. A. Each row. B. Each column. C. Rows and columns. D. Neither the rows nor the columns. The correct answer is each column. Two considerations arise in trying to tune the PSSM so that it adequately represents the training sequences. Which of the following is not their description? A. If a given column in 20 sequences has only isoleucine, it is not very likely that different amino acid will be found in other sequences with that motif because the residue is probably important for function. B. If a given column in 20 sequences has only isoleucine, it is very likely that different amino acid will be found in other sequences with that motif because the residue is probably important for function. C. If the number of sequences with the found motif is large and reasonably diverse, the sequences represent a good statistical sampling of all sequences that are ever likely to be found with that same motif. D. Another column in the motif from the 20 sequences may have several amino acids and some amino acids may not be represented at all. The correct answer is if a given column in 20 sequences has only isoleucine, it is very likely that different amino acid will be found in other sequences with that motif because the residue is probably important. Which of the following is not a feature of editors and formatters? A. Provision for displaying the sequence on a color monitor with residue colors to aid in a clear visual representation of the alignment. B. Recognition of the multiple sequence format that was output by the MSA multiple sequence alignment program. C. Maintenance of the alignment in a suitable format when the editing is completed. D. Disallowing shading conserved residues in the alignment. The correct answer is disallowing shading conserved residues in the alignment. If the data set is then unless the motif has amino acids in each column, the column frequencies in the motif may not be highly representative of all other occurrences of the motif. A. Small distinct. B. Small almost identical. C. Large almost identical. D. Large distinct. The correct answer is small almost identical. Analysis of S for conserved blocks of sequence leads to production of the position-specific scoring matrix. A. True. B. False. C. D. The correct answer is true. GDE Genetic Data Environment provides a general interface on UNIX machines for sequence analysis, sequence alignment, editing, and display. A. True. B. False. C. D. The correct answer is true. Two commonly encountered examples are the Genetics Computer Group SMSF format and the CLUSTALW ALN format. A. True. B. False. C. D. The correct answer is true. Even if many pseudocounts are added in comparison to real sequence counts, the amino acid frequencies will not have any effect or influence. A. True. B. False. C. D. The correct answer is false. MACAW is a local multiple sequence alignment program only. A. True. B. False. C. D. The correct answer is false.